the structure? What about the structure of antibodies? So the antibody is generally Y-shaped, as you've seen. It is Y-shaped. It's made up of two identical heavy chains. Okay, they are just polypeptide chains. It's also made up of two identical light chains. They are polypeptides, of course. So antibodies or immunoglobulins are proteinous in nature. Then, of course, it is composed of disulfide bonds. These disulfide bonds connect the heavy and light chains in regions we are calling domains. Okay, and then these antibodies would have the Fab fragment of the Fab region. This is the region that is able to bind antigens. Okay, so that the FAB region or the FAB fragment is one that binds the antigens, and it's composed of one constant and one variable domain on each of the chains. It also has the FC region, and the FC region is on the base of the, of course, the antibody. It modulates immune cell activity. It's composed of two heavy chains, and it's the one that determines the IgG isotype. Okay, it determines the IgG isotypes. As you've seen, we have 4 isotypes of IgG. So we can only determine the isotypes of IgG depending on the FC fragment of an immunoglobulin. So generally, this is how they look like. So this upper part, the part that is branched, the part that is branched is what we refer to as the Fab region. This is the region where antigen interaction is happening with the antibody. So this is the region where the interaction is happening. As we said, we have a heavy chain, two heavy chains, these that are dark, and we have the two light chains. Two light chains, okay? Then we have the variable domains. Variable domains, so this is the variable domains on the heavy chain. This is the variable domain on the heavy chain. This is the constant domain on the heavy chain. Then this is the variable domain, this outer part, the variable domain on the light chain. And this is the constant domain on the light chain. So of course, as you said, we have one constant and one variable domain. So if you see C, it means the constant domain. V means the variable domain. H means V, the heavy. L means the light. So you see, we have two light chains, as you've seen, and these two light chains are just present in the FAB region. The FC region only contains the heavy chains. So the FAB region contains both the heavy chain and the light chain. And the heavy chain would have the variable domain, as well as the light chain will have the variable domain. Still on the FAB, we have a constant domain, and the constant domain is almost here. Then this is where we have the disulfide bonds. This is the salt bridge here, the disulfide bond here. Or the disulfide bonds. These are the disulfide bonds here. This is the domain where we have the disulfide bonds. So they are binding the heavy and the light chains together. Okay, so ideally this is how any immunoglobulin looks like. We've seen that IgE, IgA, IgD, IgG, they are all monomeric. They are all monomeric, although IgA kind of is dimeric. If you remember IgA, there's also some disulfide bond that binds. It's not a disulfide, but it's a J chain that binds this to another. So there are two of these, like in the opposite direction. Yeah. So it's more of a di. It's a di. But IgM is pentameric. If you remember, IgM is pentameric, and the rest are monomeric. apart from IgA, IgA, because IgA is also joined by a J chain. We have two of these, so it's dimeric. Okay, so ideally that is how antibodies look like. It's structured generally. And say that antibodies or immunoglobulins, the immunoglobulin super gene family is the one that then produces the B cell receptor as well as the T cell receptor. So it means that the B cell receptor looks like this. The T cell receptor also looks like this because they're just being generated from the same type of genes. And what we call there is of course non-homologous recombination that is happening. So there's variation and mutations within those regions so that they are able to differentiate. Actually, in class switching, that is what is happening. So in class switching, there is introduction of mutations within the immunoglobulin supergene family. I just didn't want to go into those mechanisms or cascades so that then there is class switching. If mutations do not occur, then we cannot have a switch from IgM to IgG, for instance, or IgG to IgM to IgA, or IgM to IgE. That cannot happen. So there needs to be mutations within the genes that code for these particular immunoglobulins for them, therefore, to realize a change. So we also need to understand what we call by antibody responses. We need, so what are antibody responses? Antibody responses would be the antibodies that are then being produced once the B cells encounter. And these are either polyclonal or monoclonal. We talk about polyclonal antibodies because these are multiple antibodies targeting different parts of an immunogen. Okay? They are targeting different parts of an antigen. They are targeting very many different parts. So they are referred to as polyclonal. But monoclonal antibodies are just a single population of antibodies targeting a specific epitope. Okay, they are targeting a specific epitope, and therefore they're able to produce just a single clone of antibodies, a very single clone of antibodies. So we have those two types. And I'm sure as you continue experiencing in your practice, you'll see that monoclonal antibodies are very important therapeutic components. Most of the drugs, especially for cancer patients or drugs that are used for autoimmune diseases, most of them are monoclonal antibodies. They are very specific to specific epitopes of immunogens on either the cancers or the changes that are brought about in cells due to autoimmune diseases. So monoclonal antibodies are very important forms. One common example is like the antisera that we use in determining your blood groups, the ABO. So if you want to do blood grouping and tell whether you're, first of all, are you rhesus positive or negative, or are you in blood group O, A, B, or AB, what you usually do is use the antisera. So you use anti-A, you have anti-B, and you have anti-D. You only need those three antisera. Anti-D for rhesus factor, anti-A, of course, for blood group A, anti-B for blood group B. Of course, so you don't need O because in O, would somebody explain to me what is happening in blood grouping? So, yes, Polycarp says A has A, B has B, AB has both, O has none. Okay, so that is the case. So when you have antisera, these are monoclonal antibodies that are specifically against the antigen. So anti-A, it means it is an antibody against antigen A. Anti-B is supposed to be an antigen against, and I mean an antibody against antigen B. And then you need anti-D, anti-D. So you just need 3, anti-A, anti-B, and anti-D. No, so anti-A, if there is cross reaction, between, say, if you're using anti-sera A and there is cross reaction, it means that you do not have that antigen. Okay. Similarly, if you have B, you're trying anti-B, sorry, and there is cross reaction, it means you also don't have that. Okay. But then, if there is a cross reaction, I mean, there is no cross reaction in both, then it means you have both. So those, and if you have a cross reaction in D, it means that you do not have the rhesus factor. That is what it means. So that is how blood grouping is done. Just on that, you need the antisera and a white tile. That's all you need, and you're able to tell your blood group. It's not a very complicated process for you to do. So then those are monoclonal antibodies. They are very specific to the red blood cell antigens that are present. So all you need is the antisera and of course a blood sample. And then you're going to react with them. You'll be able to see and be able to classify. Although you need to be keen because some are late reactors. So you'll find that probably it takes a bit of time before the cross reaction happens. So you need to be keen before you record. So you give it at least 10 minutes for you to be able to confirm whether you are a certain blood group or not. Yeah, so monoclonal antibodies are important, especially in therapy, cancer therapies. If you see any drug that has a name, whatever it is, but it ends with MAB, we call that a suffix, MAB. It means monoclonal antibody. That drug is a monoclonal antibody. Okay, so that's the case. And now let us look at the roles of antibodies. What do antibodies do? We know that antibodies, of course, are supposed to neutralize antigens. That is majorly what antibodies do. We're also seeing that antibodies can be used for therapy. Using a technique called hybridoma technology, you're able to produce very many monoclonal antibodies against specific epitopes of various conditions, and you're able to use them for therapy. But let us see what they do in the body. So we don't want to talk about the monoclonal antibodies that are therapeutic outside the body, the ones that we induce. We're talking about the natural monoclonal the natural antibodies, not monoclonal, both polyclonal and monoclonal. So first, they promote phagocytosis. And they promote phagocytosis by doing various things. First of all, antibodies are able to directly bind to phagocytic cells. And once that happens, whatever has been bound can be phagocytosed. Okay, so they're able to bind directly, and whatever has been bound is phagocytosed. Then it's possible to form antigen-antibody complexes, and once antigen-antibody complexes are formed, then phagocytic cells are able then to phagocytose. Then it's also possible to form antigen, antibody, and complement complex. Remember, we talked about complements being opsonins. If you remember complement that we talked about being C3b. C3b is a very good opsonin. We also saw C4b is a fairly good opsonin, if I'm not wrong. Is it C5b or CC? C3b, of course, definitely is an opsonin. Okay, so just remind me whether it is C5b or C4b, one of the two. It will be a very good opsonin they caught. So complement antibody antigen complex are signals for phagocytosis. And of course, you remember opsonins, antibodies usually also are opsonin. Antibodies are generally opsonins. Complements, we have no issue. We know that complement we covered that. We were able to really, yeah, C4B and C3B are opsonins. C4B and C3B, they are opsonins. So complement is an opsonin. Antibodies generally are opsonins also. So antibodies are able to coat cells that are supposed to be phagocytosed. So antibodies play a very key role, either directly by coating the cells that are supposed to be phagocytosed or by forming complexes, and this complex could be antigen antibody complex or antigen antibody and complement complex, or the very antibodies can still directly bind cells that are supposed to be phagocytosed. In other words, they are just being opsonins in one way or another. So that is one role of antibodies in the body. Another role is that they are signaling mediators. They signal mediator cells to release immune mediators. So they signal cells, the immune cells, to release immune mediators. Now, an example here is IgE. We know what IgE is, and IgE attaches to mast cells and basophils. Mast cells, of course, is a type of white blood cell. Basophils is also another type of white blood cell. Through the Fc fragment, we know all the Fc fragment, we know how it looks like now. And the attachment is done by the Fc receptor 1. So it's called the Fc epsilon receptor 1 binding. When this happens, there is a biochemical cascade that is generated. There is a biochemical cascade that is generated. Okay, and what happens is that at least two membrane-bound IgE molecules, together with antigens, will cause increase in cyclic adenosine monophosphate. This is a messenger. Have an idea of what signal transaction. AMP is a secondary messenger. You have things like IP3 are secondary messengers, calcium secondary messengers. What they do is just amplify a signal. So decreasing CAMP, if a secondary messenger is supposed to amplify a signal, okay? But you're saying if IgE binds to mast cells and basophils, there is decrease in a secondary messenger such as CAMP, it means it is now slowing down the signal. It's not amplifying the signal, it's doing the opposite. When this happens, it leads to bronchoconstriction. And when bronchoconstriction comes into play, that is how allergic responses are then felt. Of course, we're going to discuss this in details when we'll be looking at hypersensitivity. We're going to discuss this. Now, if you are asthmatic, this is your main problem. If you are asthmatic, this is what happens actually. So we said that IgE is mainly in the respiratory tissues, produced mainly in the respiratory and sometimes in the tissues, sometimes, not all the time, sometimes in the tissues. So if somebody is asthmatic, if somebody has maybe rhinitis, that's another allergic reaction, they are overproducing IgE. They are overproducing IgE. So they're just responding to allergens by overproducing IgE, this antibody IgE. And when IgE binds to mast cells and basophils, is then going to cause bronchoconstriction. And that's why you find allergic attacks. Not asthmatic attacks are characterized by individuals having problems in their breathing. So you find that they are unable to breathe because of this. Okay. So I'm reminded of an experiment that was done. And so if you have worms, in your body, what is happening is that, again, the worms trigger production of IgE. They trigger production of IgE. And once they trigger the production of IgE, of course, if IgE is being produced in mass, then it's going to bind mast cells and basophils. Now, if you have worms in your body, you behave sometimes like you have a cold, sometimes. But classically, if you were to look at a sheep, for instance, sheep are the most common victims of this. If you want to know that a sheep is infected with worms, you just look at its nose, and if it's behaving as if it has a cold, then you know that that sheep has worms. Now, if you quantify the IgE levels, in your body, if you have worms, versus an individual who just has asthma, the person who has worms has even 1,000 times more IgE levels compared to an individual who has asthma, who is overreacting to some allergen. But why is it that asthmatic attacks can kill, while worms, in that case, will not cause an individual to end up dying just because they are bronchoconstricting? Okay, so let me just try to answer this. It happens to be an experiment that is still going on. When somebody is infected with worms, I think Miguel was on the right track in trying to answer that. When somebody is infected with worms, so you see, remember, this is a host. It's a host. So usually parasites do not want to kill the host. The host has to survive for the parasite to survive. If the host dies, then of course the parasite dies. So worms have found a way of, in as much as they are triggering the body to generate as much IgE as possible. And remember, if IgE is produced, it's just going to still attach to mast cells, it's going to attach to basophils, and when it attaches to mast cells and basophils and mast cells, they're going to burst. As they burst, they are going to release their granules, and those granules are going to cause effects in the body, including bronchoconstriction. The only thing that happens for people who are infested with worms is that they produce a very high level of T regulatory cells. They produce a very high level of T regulatory cells. And you remember what Tregs do? Tregs, other than producing the molecule called FOXP3, they produce interleukin 10. And interleukin 10 is anti-inflammatory. Now, in this case, IgE is causing inflammation to go on and bronchoconstriction. It's leading to bronchoconstriction. So IL-10 will neutralize that effect so that then these individuals do not end up behaving like those individuals who have cancer. Who have not cancer, but asthma. But if you are to take the titers of IgE from an individual who is infected with worms and put that titer of IgE in an individual who has asthma, they die immediately because the levels are just too much. So that experiment is going on, and there is a hope for people who are asthmatic that if a way is found where interleukin 10 levels can be raised in their bodies, then probably they should never worry about asthmatic attacks. They should never worry about asthmatic attacks. Okay, so that is just a by the way, but thinking we are familiar with the hygiene theory. The hygiene theory. So, of course, the hygiene theory states that people who have people who end up in environments that are dirty, they trigger their bodies, or they gear their bodies towards Th1 kind of responses, while people who are in very clean environments usually trigger their bodies or their T cells towards Th2 kind of uh, immune response. And therefore, if you're not asthmatic, for instance, you must have been exposed to that. Okay, so likelihood of you suffering from hypersensitivity is less likely, while individuals who say are suffering from maybe those conditions are triggered by Th2 kind of response. And that's why it's a very, very serious issue, especially in the developed world. The developed world during, is it called the spring? During spring, it is very serious. Very many people are hospitalized because of asthmatic attacks, because of rhinitis. People are really hospitalized because of that. So the argument here is the hygiene theory. This part of the world, maybe we expose so much to very many infectious agents, and as we expose to very many infectious agents, our bodies are more geared towards dealing with better dealing with infectious agents. Of course, that is the reason was given during the times of COVID. That the reason why Africa was not so much affected by COVID. It's because we are more geared towards Th1. Um, though maybe not true. The only reason for me that made sense is that the African population is still young compared to the European and the developed world population. And as you saw, COVID was mainly affecting individuals who are elderly, uh, and individuals who are, of course, when they are elderly, they are likely to suffer other conditions, including lifestyle diseases, diabetes, and so on and so forth. That's why they died so much. But Newton Otieno anasema ni sasa tusijifanye wasafi sana. Okay. Oh, Newton, that is it. That's your thought, Newton. I don't think Luos can ever suffer from asthma anyway. So wako na malaria mingi huko kwao. It's likely to. Yeah. Anyway, let it not sound to be tribal. It's just a joke anyway. What else? Yeah. Of course, antibodies are involved in killing target cells, and this would be via natural killer cells. You remember we talked about natural killer cells. We also talked about cytotoxic T cells, CD8s. They behave the same way. Cytotoxic T cells and natural killer cells, they have granzyme, they have perforin, and we know what that does. So, natural killer mechanisms, antibodies participate in natural killer cell activity through either direct killing, okay, via innate immune system, or via ADCC, antibody dependent cell cytotoxicity. Antibody dependent cell cytotoxicity. So this is part of the. So I said we have four types of hypersensitivities. We have four types of hypersensitivities. And one of the type of hypersensitivity that does not involve cell is called ADCC. So we'll discuss this in details when we'll be looking at hypersensitivity. Hypersensitivity is just one topic away, so should be able to cover that. Then signaling phagocytes and natural killer cells to kill target cells. That is also the other thing that antibodies do. So they're able to signal natural killer cells. They're able to signal even CD8 cells to be able to kill uh, even phagocytes, to be able to eliminate cells that are not supposed to be there. And that includes sometimes leading to autoimmunity. We'll discuss autoimmunity in details. Let us not bother about it right now. Cytokines play a key role, of course, in humoral immunity. We've seen that interferon gamma plays a key role in especially cell differentiation and proliferation. We've seen that IL-4, IL-10 play a key role in ensuring that class switching is happening, and therefore they play a key role in this. They would be important in growth, in differentiation, in activation, as well as inhibition of immune cells. So inhibition of immune cells so that for instance, interleukin 10, it will be anti-inflammatory. So it inhibits some of the pro-inflammatory cells so that then the immune response is not inflamed to cause issues. Right. So we can stop there, and maybe I would welcome one or two questions. I'm seeing a question here. Sharon is asking, what triggers class switching, or how does it come about? Okay, so I tried to explain this, and I was saying, first of all, the first immunoglobulin that is being generated is IgM. Once IgM is generated, and we know that now this infection is in the respiratory system, and the respiratory system requires IgE for us to be able to eliminate. So there is what we call non-homologous recombination. In that area that we are mutations. It introduces mutations in that particular region, and once the mutation in that particular region, there is now production of an alternative. So there is non-homologous recombination. In non-homologous recombination happening in that particular region, it introduces mutations within the gene. And when mutations are introduced within the gene product becomes different from the original product. So if you are introducing a certain mutation, of course, the protein that is being generated from a mutant gene will be different from the original. And that is how then class switching is happening. It's just introducing mutations, and these mutations produce a different product. And that's why some of the reasons as we talk about, if you are a molecular biologist or if you've done a bit of cell biology or thereabout, maybe you cover that in physiology, I'm not so sure, or thereabout, because I don't think we've ever, no, yeah, a bit of it, we did it when you're looking at nucleic acids, but maybe we are more focused on the structures. So mutations are important. Mutations are detrimental in the body. They are also important. For instance, in this case, it's a mutation that will differentiate protein A from protein B. In this case, IgM from IgG, IgM from IgA, IgM from IgA, and so on, and even IgD. So it is just mutation. Triggered by, first of all, the type of antigen, and second, the location where the antigen is.